You're listening to Conferences Online Allergy from Children's Mercy Hospital in Kansas City, Missouri. Today is February 10th, 2025. Our topic is Abbas Chapter 16, Immunity to Microbes. This is presented by Dr. Nikita Raje. She is an Associate Professor of Internal Medicine, Pediatrics, and is the Chief in the Section of Allergy and Immunology at Children's Mercy Hospital.
We're going to be talking about Chapter 16. Today is February 10, 2025, Immunity to Microbes, chapter from Abbas. I'm Nikita Raje from Children's Mercy, Kansas City, University of Missouri, Kansas City. So we went through some Jeopardy questions, and now we'll go ahead and talk about some of these concepts for each of these microorganisms. Here are my disclosures, nothing relevant to the talk today. Here's the outline. We're going to go over each of immunity to each of the type of microbe. In general, thinking about that, for each of them, we'll talk about innate immunity, then adaptive immunity. We'll talk how these microbes succeed in evading the immune system, and then in the process of that immune response, sometimes it causes injury to host cells. So we'll talk about those for each of them. So let's talk about the factors that affect the outcomes of infections. So there are some host factors and some pathogen factors. So host factors are how is the immunity of the host, innate immunity and adaptive immunity. We talk about how the specialized immune responses, for example, for Th subsets, does the immune response, is the host factor more prone to have injury from the immune response and any kind of inherited or acquired immune defects. Pathogen factors depends on the entry of the pathogen, whether it's going to cause colonization or invasion. Is it able to evade the immune system and get entry or get its pathogenic effect on the host cells, resistance to the effector mechanisms of immunity, and then whether it's able to establish latent or persistent infection, and then tissue injury or functional impairment that it can cause. So I think this is a really important chart to kind of memorize extracellular and intracellular microbes. Each of them are two different types. For extracellular, either the infection could be on the epithelial surface or it could be in interstitial spaces or blood or lymph, right? So not associated with the cell at all. And then intracellular, it could be in the cytoplasm or it could be inside the vesicles. And based on where the microbe is, our immune system identifies these sites as different places where the immune response differs. So you can see in the last row how for extracellular microbes, humoral immune response plays a big role versus intracellular, the cell-mediated immune response plays a big role. Of course, there are innate immune mechanisms that are mentioned there as well for each of them. All right, so let's get started for the first type of microbe, extracellular bacteria. So we saw these sites, whether it's in the lumen of certain tract like airway or GI tract, or connective tissue or blood or secretions. And typically, what happens when these extracellular bacteria infect the tissues? There is inflammation that causes the tissue destruction. There are toxins that can be produced by some of these extracellular bacteria, which could be endotoxins or exotoxins. Remember, this is all from med school. We have gone through this, but it's been a long time that we don't talk about these things as much. We talk about the immune response a lot, but we don't talk about the microbes as much. And so it's always a good exercise to remind ourselves of how the pathogenesis works for some of the microbes that our immune system is dealing with. So endotoxins can be something like that come from the bacterial cell wall, something like LPS in terms of gram-negative bacteria. Exotoxins are secreted. These are have different mechanisms that they use, such as they can be cytotoxic. Diphtheria exotoxin can cause can halt the protein synthesis. Cholera interferes with ion and water transport. Tetanus can cause inhibition of neuromuscular transmission, and anthrax can disrupt the critical biochemical signaling pathways. So you can kind of think about all these disorders, what these infections cause, and see how the pathogenesis work in terms of cholera causing diarrhea versus tetanus causing neuromuscular issues, and so on and so forth. These exotoxins can interfere with cellular functions and may not have to kill the cells, but they'll still interfere with the normal function of these cells. They also stimulate production of cytokines, and the cytokines themselves can also lead to some of the pathogenesis that's seen in the disease. Just showing the bacterial cell wall, gram-positive, that has a large layer of peptidoglycan above the cytoplasmic membrane, versus and has some of these molecules like teichoic acid or lipoteichoic acid that play a role in pathogenesis, versus the gram-negative bacteria, where aside from the peptidoglycan, there is that whole layer of lipopolysaccharide or LPS that we know plays a role in pathogenesis. All right, so we talked about pathogenesis. Now we talk about the immune response. First, we talk about the innate immune response. So when there are extracellular bacteria that our body comes across, there will be complement activation. So think about innate immunity, and those pathways of complement are alternative pathway and MBL pathway that don't take help from the humoral immune system, versus the classical pathway that's almost like a part of the adaptive immune system. So here we are talking about alternative pathway. The C3 takeover continues, and that they can, if they come across some kind of microbial pattern like LPS or peptidoglycans, it will get activated and form C3 convertase and then C5 convertase and form the membrane attack complex. Similarly, MBL pathway, if it finds MBL, mannose binding lectin, it can lead to opsonization, membrane attack complex. It can also recruit and activate leukocytes. So that's your complement activation in response to extracellular bacteria. So this is all kind of just a revision of what we have already talked about in different chapters, but now we are taking all that knowledge from the first 13 chapters and putting it in context to each of these microbes. Activation of phagocytes and inflammation. So phagocytes, macrophages, and neutrophils will use their surface receptors to recognize the extracellular bacteria. They are gonna go ahead and phagocytose those microbes, and then they can use, phagocytes use FC receptors or complement receptors to recognize those opsonized extracellular bacteria that are opsonized by either complement fragment or immunoglobulins. And then they're going to phagocytose those. They can also release cytokines that will bring in other leukocytes in to help to defend against that microbe. That can be from phagocytes, that could be, those cytokines can be coming from the dendritic cells as well. That all of that, those cytokines can lead to inflammation. ILCs are different types, type 1, type 2, type 3. ILC type 3, these are similar to our Th subsets, which are part of the adaptive immune system. ILC3s are similar to those, which means that they release IL-17, 22, GM-CSF. And what does that do? So that is helpful to recruit neutrophils, right? So more phagocytes, and they also help in enhancing epithelial barrier, so help with that innate immune response to extracellular bacteria. What about adaptive immunity? So the major protective immune response to extracellular bacteria has to be humoral. And we have looked at this figure. How does that work? Bacteria are detected by the immune system or the innate immune system. That message, they get opsonized maybe, and then they come across the B cells. B cells, BCR gets attaches to that antigen along with the co-receptor. Co-receptor has the complement recess or CD21, and it can be attached to the complement opsonized part of the antigen coming from the extracellular bacteria. The end result is production of antibodies, helps with the classical pathway of complement activation, and that can lead to all of the humoral immune response mechanisms like neutralization, opsonization, phagocytosis, inflammation, and lysis, all of some of these coming from the complement activation. That's for the humoral immune response. Then we talk about the, okay, so the second part here is the T cell response. So that comes from the Th17 type of response. So that causes an inflammation. Interferon gamma that's released will help in activation of macrophages, so more phagocytosis. And then the cytokines that are released can also help with the antibody production. All right, we talked about the immune response. Now, the immune response itself can also lead to some of the injury. And so the inflammation that comes from the phagocytosis or the phagocytes. So neutrophils and macrophages typically cause self-limited inflammation. They also release cytokines that will include acute phase reactants being activated, and that will cause the systemic symptoms. Cytokines can be released that are inflammatory in nature, IL-1, IL-6, TNF. These are the main cytokines that cause inflammation and fevers. So kind of, those are there with these extracellular bacteria. Along with that, there will be IL-12, interferon gamma, that can also help in activating more macrophages. It can lead to SIRS, systemic inflammatory response syndrome, or septic shock. And late phase injury from the immune response can come in terms of antibodies that cause complications such as group B strep, and for the late phase it can cause rheumatic fever or nephritis. We talked about superantigens, how superantigens are actually antigens that instead of being able to cause a antigen specific T cell activation. Remember, the TCRs for each of the clone of T cells are different, right? These are the different specificities that you can see for the TCR. So typically, an antigen will bind to one of these TCRs, and depending on which clone is activated, those T cells will get activated and proliferate to make a bigger clone of that specificity, but the rest of the T cells will continue to stay in inactive form. However, a superantigen is able to bind to that overall structure so that it can bind to the TCR of various specificities and lead to polyclonal activation, leading to widespread inflammation and cytokine storm, which can be injurious to the host. Now, these bacteria have gotten smarter and they learn to avoid the immune system or kind of, defeat it so that the pathogen can take over and cause the disease state. So for various microbes, we know that there are different ways that they use to evade the immune system. So some of the examples include antigenic variation in terms of Neisseria, E. coli, Salmonella, where some of these microbes can there is just variation in the antigens that they can carry. There can be inhibition of the complement activation by several bacteria that have learned to evade the complement system. Pneumococcus, Neisseria, all these encapsulated microbes are harder for the phagocytes to capture. So they are resistant to phagocytosis. And then there are some of these microbes called catalase positive bacteria that can still cause that can easily get access and cause infections by scavenging the reactive oxygen species. So that's your typical scenario with chronic granulomatous disease, where there's no oxidative capacity of the phagocytes that can kill these microbes. So there are these catalase positive microbes that are able to degrade the hydrogen peroxide that our body can use, or phagocytes can use, from the bacteria themselves and kill them. But the catalase can disintegrate that hydrogen peroxide into water, and then those microbes can still cause infections. All right, moving along, we'll talk about immunity to intracellular bacteria. We know that the intracellular phase needs cell-mediated immunity. We'll talk about innate immunity and then adaptive immunity. Innate immunity again includes phagocytes. There are various ways that are used to recognize the intracellular bacteria. These could be toll-like receptors, nod-like receptors. They might recognize patterns that are specific to these intracellular bacteria. And then they can also have double-stranded DNA that can be detected by the STING pathway. And then use that to recognize the microbes and then undergo phagocytosis. There are dendritic cells and macrophages that will release cytokines that will help in immunity against the intracellular bacteria. NK cells play a big role. Bacteria activate them by inducing some of the activating ligands on the infected cells, and then the NK cells can also produce interferon gamma that in turn activates the macrophages. So NK cells and macrophages, dendritic cells, all kinds of phagocytes, so macrophages and neutrophils, all of them play a role here. Talking about adaptive immunity, we know that cell-mediated immunity plays a big role. So Th1 type of cells get activated. There are times when Th2 type of cells also play a role. They have their own cytokines, Th1 producing interferon gamma, TNF, and help in activation of macrophages. Th2 can produce IL-4, IL-13, use the classical macrophage activation or alternative macrophage activation, and regardless help with these intracellular bacterial infections. Injury from the immune response to intracellular, there's a lot of macrophage that gets activated, can cause delayed type hypersensitivity. The intracellular bacteria will resist killing within the phagocytes, so they can persist for a long time, bring in other players or other cells that can cause inflammation, that can lead to formation of granulomas. It prevents spread of the infection, but at the same time it can cause functional impairment of that organ due to tissue necrosis and fibrosis. All right, so some of the examples that are given here: tuberculosis that causes caseating granulomas, and you also see the PPD when it's placed, it causes that small reaction. All right, immune evasion by intracellular bacteria. Again, some of the um, intracellular bacteria can evade the phagocytosis or phagolysosome formation, such as mycobacteria, legionella, that can actually escape from the phagolysosome. There can be inactivation of reactive oxygen species or nitrogen species in terms of macrophages, like in leprosy. And that's listeria that actually can escape from the phagosome, and then they can get into the cytoplasm instead of being in that phagolysosome. So they can escape the phagocytosis or killing inside the phagocytes. Moving on to talking about fungal infections, there are different types of fungal infections. So there are mycoses that are endemic to certain environments. And in those cases, there can be infections like opportunistic infections, and we'll talk about some of these. But anyways, so the host factors that can also play a role, such as whether there is good immunity or if there are immune defects, and particularly in case of fungal infections, if there are any kind of mechanisms or immune suppressive situations. Fungal infections are more common, such as in case of chemotherapy or neutrophilic defects, if there is bone marrow suppression, in case of being on conditioning regimen or having HIV. All these can lead to defects in immunity to fungal infections. Talking about innate and adaptive immunity to fungal infections. Innate immunity, think about neutrophils, macrophages, so phagocytes can play a role. They use their pattern recognition receptors, such as tool-like receptors, and of course dectins that are specific to fungal infections, that help them recognize these fungi. And then adaptive immunity, we know that the major response comes from the cell-mediated immunity. So CD4 cells and CD8 cells, CD4 cells cause Th17 kind of response if it is extracellular, and if this is intracellular, then you'll have more Th1 response. And then there is a rule for adaptive immune response, the antibody response, not as well defined. Immune evasion, Cryptococcus, for example, it can inhibit the cytokine production by macrophages. So the inflammation that's caused by these cytokines like TNF and IL-12 will be missing. And then they can stimulate production of IL-10, which is anti-inflammatory, so it inhibits macrophage activation. So that's how it uses or tricks our immune system and evades it. Injury, the immune response to fungal infections is Th1 response, so it can cause granulomas such as in histoplasmosis. Moving on to viral infections and immunity to viruses. Remember, viruses are obligatory intracellular microbes, in that they can either cause a lytic infection, or it can stay latent, which means it's gonna stay dormant. Talking about innate immunity to viruses, the most important mechanism is production of type 1 interferons. These type 1 interferons, if you recall, come from these cells called plasmacytoid dendritic cells. And we have several different pattern recognition receptors in various places, on the cell surface, or more in the endosomal area where there are toll-like receptors. But apart from being attached to cells or being in the cytoplasm of cells like Rig-like receptors in STING pathway, there are um, um, other pattern recognition receptors that are present, or molecules that recognize patterns in the extracellular places as well. Type 1 interferons inhibit viral replication in infected as well as uninfected cells. We saw the mechanisms, how it warns other cells, adjoining cells, so that they don't get infected. And then we know that NK cells, natural killer cells, play an important role in viral infections as well. What happens is when the virus affects a cell, class 1 MHC expression is shut off. So there is no class 1, say this is a liver cell, and there is no, because it's affected by a virus, there is no class 1 MHC on the surface of that cell. Now, natural killer cells know where to go and find the cells that don't have class 1 MHC on them. And once NK cells get activated and there is a missing class 1 MHC on the surface, they know that that's the right target and they'll kill that cell. That's how the NK cells work. And then apart from the innate immunity, we know for the viruses, there are both antibodies as well as cell-mediated immunity that's important. How are the antibodies going to play a role? Viruses, we know, are intracellular, but during their extracellular stage or phase, where they are released from a cell and before they infect the adjoining cells, is where the antibodies can work. And they work in different ways. For example, there can be neutralizing antibodies, which means they are just going to attach to the virus, and they are going to prevent the entry of that virus in the adjoining host cell. They can also help in opsonization and complement activation. Unfortunately, once they are inside the cell, the antibodies have no control over those viruses. So they cannot eradicate the infection just because they cannot get to the intracellular reservoirs of these viruses. So for that, you need CD8 cells. So CD8 cells are absolutely essential to eliminate those viruses. And then these viruses the CD8 cells can help in targeting all kinds of infected cells, and then sometimes they use cross-priming or cross-presentation for these viruses as well. All right, talking about latent infections. So kind of keeping in mind that when the virus can establish a latent infection, which means they are not going to be multiplying and they're still going to stay dormant in the human body. But when the immunity wanes, there can be a reactivation of that latent infection, and that virus can get into the lytic phase again and cause cytopathic effects. One of the important virus or example of this latent infection is to remember in viruses like EBV and other herpes viruses that can cause latent infections, such as zoster after a chickenpox infection. All right, talking about these latent infection versus here are these innate immune responses, type 1 interferons and NK cells. And then over time, they need cell-mediated immunity, CTLs, and antibodies that will come into effect later part of the viral infection. Injury caused by viruses. Some viruses can infect the cells but do not cause direct cytopathic changes, right? So one of the example of this is hepatitis. The virus itself can infect the liver cells. Hepatitis virus can infect the cell, but they don't cause any problem. But unfortunately, once the virus is detected, the immune response to that virus can cause cell injury. That's hepatitis B, where the CTL response itself is more damaging than the virus itself. Another example is antibodies that deposit in blood vessels and cause systemic vasculitis. Molecular mimicry is another way the viruses can cause injury, where some viral antigens can mimic self-antigens. All right, here is a long list of microbes and how they evade the immune system. We know flu virus that shows antigenic variation every year, rhinovirus, HIV, they can have variation in their antigens that are displayed and evade the immune system. And that is one big reason why influenza virus vaccines have to be taken every year, so that the antigens that have changed, we get the immunity against those antigens. HSV virus, it can actually block TAP transporter, which plays a role in class 1 MHC processing. Also CMV, that can remove class 1 molecules from the endoplasmic reticulum, right? So antigen processing, that is important to present those antigens to the T cells. That process itself is affected. CMV can also use decoy MHC molecules. And remember, the NK cells are targeting cells that do not have MHC on them. So if there is a decoy MHC, NK cells think, well, there is an MHC on the cell, so the cell should not be killed. So they don't target those cells that are infected with CMV. There are some microbes that can produce cytokines that are very similar to our cytokines, such as vaccinia or pox viruses, so smallpox or other pox viruses that can produce cytokine homologues like IL-1 and interferon gamma. CMV can produce chemokines. Some viruses will actually produce cytokines that are anti-inflammatory, like IL-10 in case of EBV. HIV can actually infect and reduce the number of immune cells. There is inhibition of complement activation in terms of HIV and some of the other viruses like vaccinia and CMV. So remember, there are different regulatory complement factors such as factor H, CD59, and these viruses can play with that complement system and inhibit the complement activation. Some of the viruses can affect the pattern recognition receptors so that they are not even detected. All right, so this just basically shows the same kind of process here. HSV blocking TAP transport, removal of class 1 MHC from endoplasmic reticulum by CMV. So kind of going through this whole class 1 MHC pathway and how these viruses have learned to evade this whole system. And then antigenic variation is shown on the other side. Immunity to parasites, we have protozoa, helminths, ectoparasites. Thinking of parasites, they are different. They have a life cycle where they are in the humans or vertebrates. They can also use an intermediate host for their survival. And then humans are infected because of bites of the intermediate hosts or sharing environment with them. So this is just a long list of different kinds of parasites, helminths versus protozoa. Helminths can be nematodes, cystodes, or trematodes, and so this is something that we all learned at some point of in our careers, but kind of thinking about how these are dealt with by our immune system. So phagocytosis is one of the ways where these parasites can be dealt with. For protozoal infections, TLR2, TLR4, they can detect them, they can help in phagocytosis. Once they are inside the phagocyte, they undergo killing, like the in the phagolysosome that we know of. However, helminths are too large for phagocytosis, so at that time the phagocytes may extrude the substances onto them, such as what we learn as nets, right? Neutrophil extracellular traps. So that's your nets basically used in helminthic infection. Complement pathways also activated. Adaptive immunity, cell mediated in case of intracellular microbes. Helminths are extracellular, and antibody response is more important. Injury, some of them can cause granulomas such as schistosoma or filariasis, and some of them can cause long-term issues with deposition of complexes in blood vessels or glomeruli and cause vasculitis, nephritis, such as in malaria and schistosomiasis. Evasion, same kinds of mechanisms that are used by these parasites, antigenic variation. There can be resistance to some of the mechanisms like complement and CTLs by schistosomas, and then, inhibition of host immune responses due to filariasis that causes lymphatic obstruction. So some of those immune responses are not normal when there is lymphatic obstruction. And then sometimes some of the parasites like entamoeba will just shed the antigen, so it's no longer an issue for the microbe. All right, that